baju ukurannya. Baju mana? Pilihan baju. Sebenarnya bajunya ini kan kemarin mama kan ngomong aja di pilihan ukuran baju apa aja.
Okay. Test. Are you hear me? Halo.
Halo ya. Coba lagi, Mbak.
Tes. Sudah, Kak?
Udah masuk kok suaranya.
Oke. Maybe we can time is 4 right now. We can start the discussion. Before we start, I would like to apologize to all of you because my lamp is in my house is off. So I use the phone because my my laptop is off too. There is no.
Okay, it's totally okay.
I'm sorry, it's accident. There is an accident in the last minute.
Okay.
Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Okay, hello.
Hello.
Ya, alhamdulillah, all praises Yeah, Alhamdulillah, all praises to Allah that He still give us a chance and opportunity to be here in scientific presentation form 2. I hope you can listen my voice well. I'm so sorry. I hope you are in a good condition. Stay healthy, stay safe, and stay happy. Okay. Before we start, let me introduce myself. My name is Rizka Fitria Anggraini, and you can call me Rizka as a panelist in this section. Before we go to the sections, I would like to say thank you very much to all of you that take the time to be here. Although it's a final exam, I think it's a final exam time, but you see, thank you very much. Hope we can enjoy this section. Okay. Before we start the sections, I would like to introduce about SP. Maybe some of you still not familiar about this program. Scientific Presentations, or SP, is a program managed by R&D department of BSC that invites all of you, whether a board or a member of BSC, to present your current research or your previous research. I think it's a great chance for all of us to present our research, to get some feedback, as well as to learn speaking in English as well. And this program, this program is held twice in a month, in second and in the fourth week in every month. So if you are interested, you can fill the online form that regularly will be shared by R&D in your group. So please kindly watch the group and join as a presenter in SP. Okay, let's start the presentation. In today, we will talk about some interesting topics about food and nutrition that will be held by the two presenters. Okay, the first presenter is Kak Wildan. Let's say hello. Hello, Kak Wildan. Are you hearing me, Kak?
Hi. Hi.
Okay. Kak Wildan is a master student of food science, and he will talk about present about identification of umami compounds, ya Kak. Identification of umami compounds in fisheries resources. Okay, for Kak Wildan, time is yours.
Okay, presentation. Should I share screen, or the committee will share the PowerPoint of?
Okay.
Hello, Rista.
Yeah. I think you can share, and if you have difficult, the committee will be help. Maybe you can share by yourself.
Hi, Rista. I mean. What?
Can you hear me? Hello?
Maybe connection Mas Wildan broken.
Okay. Sorry, then maybe your connection is unstable.
Should I share screen share will here when I want decibel partition.
Oh, okay.
I'm sorry. Okay, I'm the host now, so I would like to change you to be the co-host, right? Okay, already.
3G. Hang on.
Hello, Mas Wildan. I'm sorry.
Yeah. Do
You should share screen on your PPT. Nah, yes, okay.
yeah. Is your already right?
Yeah.
Yes.
Suaranya bergelombang. Okay. Anybody can hear?
I can hear, but it's not good.
Aduh.
Have a
Anybody can hear my voice right now?
Yes, of course. It's a clear.
Okay. I'm so sorry. I'm having trouble in connection.
Okay, it's okay. Go.
Suaranya juga. Bisa nih.
Hello, I'm sorry. Is Rista lost from this connection?
Yeah, sorry. Pick and move.
Okay. So, Pinendang, I ask you to be a house now.
Okay.
Okay. Okay, Mas Wildan.
Can I start right now?
Yes, please start.
Okay. Bismillahirrahmanirrahim. Before delivering the presentation, I would like to deliver thanks to committee of this scientific presentations. In this case, is all the staff or research and development Bogor Science Club, and then the members, all participants of this scientific presentation for the volume, the second volume. My name is Ahmad Wildan, and currently an of food science and technology presentations. This is like a colloquium. It's not the result, it's not the presentation of the result of the research, but this is a research plan presentation which is titled Identification Peptides and Compounds Contributing to the Umami Test on Fermented Product. And the fermented product I use is terasi, which is produced from Faris region in Indonesia. Okay. Okay. Before I give presentations to the what is the umami, where is the main point of the presentations, let me know what is the basic test of, I mean, here I will bring you to an overview to a role of basic then differ. You can differ how different their expressions. When people express consume, for example, when they say enak or yeah, in Japan, umami is people in Japan will express themselves to say that their food is delicious, and which is umami. And umami is associated with the monosodium glutamate or MSG. We know, and it usually contained in several food products such as beef extract, soy sauce, protein, hydrolyzed, and ripened cheese. But many previous research reported that umami is not merely about the glutamic acids, or it's not about the amino acids, but umami also can be obtained from glutamic acids like aspartic acids, and also small peptides such as 5 nucleotides and several organic acids. And here is explanation about the fermentation of terasi. We just fish or shrimp as the raw materials, and to make terasi itself, and it needs is about 2 weeks for having a terasi. And you can see this is the component of terasi itself. All right, and this slide presentation source
Sorry, Mas Wildan. I'm sorry, Mas Wildan, have a trouble. Maybe your connection in internet unstable.
Do you hear my voice?
Now clearly. Maybe before slide you can repeat.
You hear my voice right now?
Yeah. Yes.
Hi. Okay, can I continue my presentation? Hi, excuse me. Hi, anybody can hear my voice?
Yeah, we can hear.
Oh,
Maybe sometimes.
so can I continue? Can I continue my presentation?
Okay, you can continue.
Ya, Kak? Yes. Sorry, kan?
Maybe connection internet Mas Wildan is have a problem. Maybe change.
So
Maybe Saleha can
answer?
the first presentation. After Saleha, Mas Wildan.
Okay. Okay. I should share. Wait. Can I start now?
Okay. Mbak Saleha can start.
Okay. Wait. Hello, everyone.
Hello.
Is my voice clearly? Do you hear me? Can you hear me?
Yes.
Clearly?
Yeah.
Okay. Itu. Halo.
Yeah. Yes, Kak. Saleha.
We can hear Mbak Saleha.
Okay.
Maybe you can start your presentation.
Warahmatullahi wabarakatuh. Speaker 2, I wish that I have to switch to as head of area department. To thanks to R&D for conducting this event. Scientific presentation volume 2. You introduce yourself about my friend and I medicinal food product that it is original that we made it when I was in the bachelor degree. So this is lactate, a yogurt of combination lactobacillus rhamnosus and polyphenol green tea extract for lowering uric acid in rat. Okay, so this is my supervisor and then my friends. Okay, and goat. What do we know about goat? Do you know? Goat is a common and complex form of arthritis. It is characterized by sudden, severe attack of swelling, redness, or tenderness in the joint. They often in the base of big toe. So in Indonesia, goat is commonly say asam urat. And good symptoms may come and go, but there are ways to manage symptoms and prevent the flares. So gout is mostly happen in the older adult and in the elderly. And as we know that prevalence gout, arthritis, or asam urat in Indonesia is 24.75%. That we can say that 1/3 of the people of Indonesia is severe or got this disease. So if we look back, the yogurt and green tea extract in Indonesia is spreading widely, and most of people know about yogurt and green tea extract. And I would like to ask to you, do you know yogurt, right? Or green tea is very common in Indonesia. So from those things, my friend and I trying to make the combination, the yogurt and then the green tea extract that have effect to lowering the uric acid level. So we make lactate. Lactate is bring easily, and then you bring that yogurt so feel lactate. You can say that I'm like, so this is a research product, and I would like to, because this is my research. Lactate is base lowering xanthine oxidase. So then and produce the uric acid. Oxidase, we are able to lower it. The yogurt, which is able to lowering the uric acid levels. It's familiar, and then maybe taste like me that I so this lactate may low reduce the fat content and lactate. And the goals of this research one is to analyzing about the effectiveness level, and then the second is analyzing foods are making any medicinal food product alternative for good international or national to give green tea extract, grinded green tea. Then we filter it into the tea mat, and also we get the from the extraction, and then we mix again. And by rotavapor, and also temperature, and also the green tea extract. With the inoculate of the bacteria, so we use inoculate the bacteria first, and then we mix the inoculated bacteria with this skim milk sterilisasi gitu, pasteurized of milk, and then the
Sorry, Mbak Saleha.
Sorry, yes.
Your voice is not clearly something.
Okay, so also should I repeat it?
Yeah, you can repeat
from this yogurt method, right?
Yeah.
Okay. This is how I and my friends make the yogurt. So first of all, we use the Lactobacillus rhamnosus bacteria, and then we inoculate NDMRS broth, and then we add the inoculated bacteria to the skim milk that have pasteurized, sudah dipasteurisasi, and then after we mix it, we ferment it in the like incubator within 8 hours. Then after that, we get the yogurt fermented by Lactobacillus rhamnosus with the green tea polyphenol extract. So we mix it by manually procedure. So after that, we give the intervention product to the rats. So here we got 30 ml with rats, approximately 2 months, and then weigh 150 until 250 grams. And then we divide into 1, 2, 3, 4, 5 into 5 groups. That group consists of 5 rats. So we got the normal control that we didn't give any intervention on that groups. Negative control, we just add the hyperuricemic condition, and then we get the intervention group, the first lactate 1, lactate 2, and then the lactate 3. So we have 3 of. So this is the time 1 rat intervention. We did limitization, and then.
I'm sorry, Mbak Saleha.
Yeah.
Maybe you can turn off the video.
Okay.
Your signal may be not good, gitu.
Okay, and then the acclimatization. For 40 days, of chicken and uric acid level. To the intervention in the morning. So we take analyzing about after we did before, we got this result. You can see on your screen that groups of the lactate 1, lactate 3, and lactate 3 have the decreasing of the uric acid level after the induction. So, but the statistic analysis said that just lactate 2 and lactate 3 that have significantly effect. Okay, I'm sorry, because I just can share about the uric acid level, and then the other result you able to see on our published paper. Now we still on progress to publish this research. Okay, and after the research, we can say that the lactate dosage 2 and the lactate dosage 3 is able and significantly to decreasing the uric acid level in rats. And this is the documentation that while we are doing the research, making any yogurt, and then we inoculate the bacteria. And maybe it's enough for me. And thanks to you that have listened my presentation. Thanks, and wait our published paper. Thank you.
Okay. Thank you very much, Saleha. Anyway, are you hear my voice? All of you?
Yes.
Oh yeah, I'm so sorry because my internet is very poor because it's raining hard here. Okay. We go to Kak Wildan maybe. Hello, Kak Wildan.
Yeah.
Kak Wildan. Okay.
Sorry. Seriously, I'm having bad trouble here. That's why I moved from one place to another to get a better network. So anybody can hear me right now?
Yes. Yes, Kak. Your
Okay.
voice is clear right now. Yes. Okay, Kak Wildan. Maybe we
Yeah,
can start again with the presentation.
yeah, sure. Okay. Uh, Sorry for everything. Sorry for bad network I'm facing right now. So I back to the first presentation because to be honest, I forget which slides I have presents already. That's why I back to the first presentation. This is okay for you, right?
Yes, it's okay.
Okay.
Please go
Okay.
ahead.
I would like to deliver my research plan presentation. To be note that this is not the result of research presentations, but this is a research plan presentations. It's kind of a colloquium. Okay. And I bring this presentation, which is titled Identification Peptides and Compounds Contributing to the Umami Test of on Fermented Product. And the fermented product I bring here in this presentation is terasi, which is produced from various regions in Indonesia. Okay, this is an overview of role of basic tests. As I mentioned before, people when they are facing the food with a various taste like sweet, salty, bitter, sour, and umami, they will give different expressions. And umami here is just in the middle between sweet and the bitter. Then this is an overview of umami itself. Umami is, the origin from Japan word. People in Japan, when they are having good food and they want to say delicious, they say umami, and it's usually referred to the monosodium glutamate, and it has strong associations with monosodium glutamate. But based on the previous research, many researchers say that umami is not merely about monosodium glutamate or other amino acids, but can be also from such as the peptides and several organic acids. And then this is because I'm using terasi. Anybody know terasi? Uh, That's why this is the substantial of the materials how to make terasi. We need is about 12 to 20% of salt as one of the important material to make a fermented product, and then the raw fish or the shrimp. It's depend on what terasi that you want to make. But here I'm not use I want to make a terasi from the beginning step, but the terasi that I have is it's ready to eat. And for the component of the terasi itself, you can see from the slide like the water content and the protein content, and the main point is a protein content that's about 24% from 100% raw dry of terasi itself, and this is according Kusuma Wardani et al. 2013. As I mentioned before, umami source fisheries products is one of a product that it contains umami source like abalone shells, sea urchins, green crab, and squid. And this data is from the previous research that researchers have conducted research about the umami. And the sources of umami itself, as I mentioned before, is not merely about the monosodium glutamate. It's not merely about a sodium glutamate or glutamic acids, but you will have umami from several sources like peptides and then nucleotides, mixture of salt and free amino, and of course from the free amino acids like glutamates, glycine, arginine, alanine. And based on the research of Zhang et al. 2012, in buntal flesh fish, it also contains umami in the range 836 Dalton. And this is to characterize the peptides contributing the umami test of the product. So this is the main step. The first is ultrafiltrations, ultrafiltration using a membrane, 3 kilodalton, and then when it produces several fractions, it continues to do chromatography using Sephadex G15 or Sephadex G25, and then to RP-HPLC to identify what kind of the amino acids that give the most umami test of the product, and continue to the LC-SEMS to know to purificate what kind of the peptides that give or that contribute the most umami test of the product. This is the research problem. We know that many regions in Indonesia, such as Bangka Belitung, Cirebon, and then Madura, they have tracey producers. That's why this is becoming one of the problems why this research should be conducted, because every producers using different raw material, they use also different methods how to make a tracey itself. And then the characterizations like ultrafiltrations, as I mentioned before, continue to chromatography, and then using the instrument RP-HPLC and LC-SEMS to identify what kind of the peptide itself. So when we're talking about the objective and the benefits of this research, the first objective is to characterize the water-soluble extract of the sample. Okay, when I say sample, it means tracey. To characterize water-soluble extract of the sample that is potential in giving strongest umami test. And the second objective is to fractionation the water-soluble extract itself of the sample using the instrument chromatography ultrafiltrations, and then continue to isolate subfractions of umami. That after this isolating subfractions process has been conducted, then continue to using RP-HPLC preparative to characterize umami fractions using LC-SEMS. While the benefits of the research is of course to giving such an information toward compound which is contribute umami test, and it can be also used for the further investigations or further research regarding umami, and then to exposing or to empowering the potential of to empowering the product of local commodity in Indonesia to be utilized as a new sources of umami compound that can be used as a flavor into the food. While this is the hypothesis of the research, the first hypothesis is one product of tracey or one product of the sample obtained from 5 regions will have the strongest umami using sensory analysis, because we will use also sensory analysis during the research. And then the second hypothesis is fractions which is obtained from ultrafiltrations using membrane will contribute the strongest umami rather than the fractions that is obtained from the membrane more than 3 kilodalton. And then the third hypothesis is fractions of chromatography ultrafiltration will give fraction with different intensity. And the last hypothesis is to identification of peptides using instrument LC-SEMS will show peptides contributing to the umami test. While the time and the place, this research will be conducted on March 2021 to June 2021. And I will conduct the research in the Laboratory of Sensory Analysis in Universitas Gadjah Mada, and also in the Laboratory of Food Processing, Universitas Gadjah Mada also, and another laboratory in Subang, which is Balai Besar Padi, West Java for flavor analysis using instrument HPLC and LC-SEMS. this is the materials will be used during the research. You can see there are several materials for several analysis and for several identifications like natrium analysis, and then for the septic fractionations, for identification of peptide using LC-SCMS, and several analyses that will be conducted during my research. While this is the tools or the instrument will be used during the research. The first is after we got a water-soluble extract, we will conduct ultrafiltration process using membrane ultrafiltrations, 3 kilodalton with the vacuum pump, continue to the fractionations using Sepadex, and then we will have also HPLC to isolate the fractions, will give the strongest umami. And then we will use also LC-SCMS to characterize what kind of the peptide contributing to the umami test, and then sensory analysis using several tools that will be needed. And this is a step of study. The first is I collect 5 samples of terasi, varies from 5 regions from Indonesia to make the water-soluble extract itself, and then continue to fractionations process using ultrafiltration membrane, 3 kilodaltons, and then continue after finish, continue to the isolation process using RP HPLC preparative, and the last one is fractionation chromatography using after fractionating water-soluble extract, we move to the fractionation chromatography filtration, use gel Sepadex G15, and the last one is isolation subtractions of umami using RP HPLC preparative. And this is actually how to make a water-soluble extract. Anyone can look to the slide. After the product of terasi is left in the freeze dried, it's continued to the chemical analysis, such as how to analyze the pH, how to analyze the sugar content, natrium content, and the nitrogen content. While different part of the freeze dry of terasi itself will be used to sensory analysis using TDA or test dilutions analysis, and we will use several panelists to analyze the sensory of a product or of a sample. And this is the fractionations. Water-soluble extract using ultrafiltrations membrane, 3 kilodalton. The first, water-soluble extract of chosen terasi will be used. And then, we will have, in this process, we will obtain 2 fractions, the fractions under 3 kilodalton and another fractions with weight molecule more than 3 kilodalton. And then each fractions will be used to chemical analysis that I mentioned before, and also will be used to sensory analysis. And we will have the chosen ultrafiltrated or selected ultrafiltrated fractions. While chemical analysis like pH, total acid, total sugar, natrium, nitrogen, and sensory analysis, we will have test dilution analysis with the panelists, selected panelists. And this is fractionation using chromatography ultrafiltration gel Sepadex. So the selected fraction of ultrafiltrations, the product of the previous ultrafiltration will be used in this fractionation using gel Sepadex. For what? It's for, disease to analyze fractions. In this analysis, we will use 214 nanometer and 254 nanometer. And it is similar like the previous analysis. We will also have the chemical analysis and sensory analysis. And then finally, we will have a fraction umami. And this is isolations, subtractions of umami using RP HPLC preparative. We will inject the sample to the HPLC preparative, and we will have data like you can see from the slide. We will have a chromatogram to know what kind of the amino acids, and we will have also the data about the peak of the chromatogram. Then the product of these fractionations will continue to the characterizations umami using LC-SEMS. And we will have also sensory analysis like the previous analysis using panelists. And talking about the sensory analysis or TDA, test dilution analysis, we will concerns to the sensory characterizations is to know the test intensity level of sample using trained panelists with the criteria. Anyone who doesn't have any problem or who doesn't have any limitation in consuming food, and they should be passed the selections of the panelist test. It's been based on the ICO. And the first one is panelist screening first, panelist training, and continue to the sample analysis. And this is about panelist selections. We will have 50, it's about 50 panelist candidates. And then they will have a matching test and also to detections of 5 basic tests. 14 panelists will be selected to go to the next process. For the matching test, we will use several stimulus, and the stimulus is sent between the matching test and the detection test. And then the last panelist selections, there will be tests. They will have several sample with different factor of dilutions of a sample that will be used in the test. And the sample is water soluble extract of the sample. And then we will have the chosen factor dilutions and continue to the panelist training. And this is the last of analysis, that is statistic analysis. We will have one-way analysis of a variant to know differences of sample and the water soluble extract of the 5 sample as a result of chromatography and RP-HPLC. Once it's found differences between the result, then continue to the Duncan test. And then after we know which one different, t-test is used to know the differences among ultrafiltration fractions. And we will have also a data in the PCR or principal component analysis, because in this research, we use 5 sample obtained from different region in Indonesia. So PCA data is the best data to expose regarding which one the sample that has similarity in the chemical component or in the umami compound of each sample. And that's all of my presentation. So sorry if there are a lot of the mistakes during my presentation. So I'll give this session to the panelists or to the moderator.
Okay. Kak Yudan?
Yeah.
Did you finish?
Yeah.
I'm so sorry. I just go out from the Zoom and okay. Thank you very much, Kak Yudan. Okay. This is the presentation today. And before we go to the next section, is about discussion time. Maybe I would like to make some conclusions from those presentations. Kak Salihah is tell about yogurt from Lactobacillus rhamnosus and polyphenol green tea for lowering uric acid in rats. From the research, we can conclude that polyphenol green tea can lowering uric acid in rats with the proportion 15% and 30%. Right, Kak Saleha?
Yes. Exactly.
Okay, and then, hello?
Yeah. Hello, Rista. Do you still connect with us? Okay, maybe Rista is unstable connection there.
Hello? I'm so sorry.
Yeah.
Can you hear me?
Yeah, yeah.
Okay. Okay, I just go out again from this room. I'm so sorry. Okay, maybe we can start. We can go to next section. It's discussion time, right? And anyone here who have a question, please raise your hand with the raise hand button and directly speak for the question or maybe some feedback or suggestions and any others. It's not only a question, but you can also give a feedback maybe or the suggestions and others. And you are not allowed to write your answer, your questions in the chat box because we can try to try to speaking in English. And yeah, if anyone who have a question, please raise your hand and then tell your name and then the question. Okay. Before that, I want to remember that there is an absence. There is an absence and evaluation form in the chat, so you can fill from now. Okay, there is one question. I think it's Mas Irfan. So Mas Irfan, hello.
Hello, can you hear my voice?
Yes. Yes, Kak
thank you.
Irfan.
Thank you very much. I think I'm very interested in this scientific presentation because it's about food. Yeah, I like food very much. Okay, I want to ask a question for Miss Saleha and also Mr. Wildan. First one is for Miss Saleha. I want to ask a question that you combine the laptop. The first, actually, I have two questions for you, Miss Saleha. The first one, do you know where is the Lactobacillus rhamnosus? I don't know how to pronounce it. How do you know where this is isolated? You know what I mean, right? This isolate is isolated from what? Like from milk or from baby feces or maybe another source of isolation, and another source of something that you can isolate this lactobacillus species. I want to know where is this bacteria is isolated. And the second question is, do you combine the lactobacillus rhamnosus with polyphenol? But in microbiology, we know that polyphenol is one of the chemical compound that can be an antibacterial compound. It can break down the bacterial cell wall and also so it can be an antibacterial compound by destroying the bacterial cell wall. And can you explain this? Why you is there a negative effect of this polyphenol compound with this bacteria, because you combine this a very different these two different thing. Yeah, maybe you get my question, right, Miss Aleha?
Okay.
Miss
Thank
Aleha,
you.
do you get my question?
Yeah, yeah.
Okay, okay. And then I will ask Mr. Wildan. Actually, umami is a very new knowledge for me in the food science. As we know about sweet and another test, but I actually didn't know more about umami. The question is, what is the factor that wait, what can I say? What is the factor that mempengaruhi that can make us to feel this food is much umami than another food? Do you get my question, Mr. Wildan?
Would you like to please repeat your
Okay.
question?
Wait a minute, because ada pengamen sebentar. Okay, I'm sorry. The question is how, what is the compound? You say that the chemical compound that content in the food that can make us taste the umami is not about glutamate acid, right? There are so many chemical compound that can make us feel or taste the umami, right? And I want to know from that from that so many compound that you say before, what is the most the influence compounds, what is the most influence compound, what is apa senyawa kimia yang paling berpengaruh for us to test the umami test. Okay, do you get my question?
Yeah, thank you, Mr.
Okay,
Irfan, for the
thank you.
question.
That's all for me, and sorry for my bad English. Thank you.
Okay. Thank you so much, Kak Irfan, from the question. Maybe Kak Saleha can start to answer.
Okay. Is my voice clearly?
Yes. Yeah, Kak.
Okay. Well, thanks for the interesting question, Irfan. Well, that's a good question. And as we know that Lactobacillus rhamnosus is Lactobacillus rhamnosus source is from the intestine. I mean, like dari usus gitu. And prior research said that the Lactobacillus rhamnosus, which is formed or extracted from the infant intestine, is able to lowering the uric acid. But because of ethical clearance, so it is not appropriate if I extract the lactobacillus rhamnosus from the baby intestine, then made it and extracted with the food. I mean, it's not the good choice. So in this research, I buy the lactobacillus rhamnosus. Jadi aku tuh beli gitu lactobacillus rhamnosusnya. So before I apply to the yogurt, I make some rejuvenation. Jadi kayak peremajaan bakteri, something like that. Okay, is it answer? I mean, like, is it clear, or do you want to ask?
Where is the place that you buy the isolate? Is there your campus? Or
It's
Lipi?
a secret. Enggak.
Okay.
Iya, jadi aku belinya di UGM, in UGM laboratory. So in there, you able to buy many type of bacteria. But if we want to use the certain bacteria, we use we have to get the ethical clearance before we do or apply to the food or something else.
Okay. Okay, I think it's enough for the first question.
Okay, and the second question is, I don't know. I mean, like, because I'm not analyzed into the chemical compound into the micro answer that question. But if we see from the conclusion or from the result, we know that the result is able to lowering the uric acid. So this is good suggestion for my research. Maybe we able to know deeper about the chemical compound of the lactate yogurt. That's Irfan.
Okay, so you didn't know if this chemical compound can inhibit the bacteria or not, right?
No, but
But in
this
I'm sorry,
research,
I'm
but
sorry. Mohon maaf tadi pas kamu presentasi itu putus-putus. So I want to know where when you mix this polyphenol with the bacteria culture in what step?
so okay. So I want to give a brief explanation about how I make the lactate or the method. So the first of all, we made the yogurt. So we mix or we fermented the skim milk, the skim milk by the lactobacillus rhamnosus. So we get the lactobacillus rhamnosus yogurt. Then we not mix directly between the bacteria and then the polyphenol. No, we use the yogurt or we use the milk as the vesicle to meet the lactobacillus and then the polyphenol extract.
Okay.
Then we do it. I'm sorry. Then we do it all in the manually procedure.
Okay. I think it's answered the question because in the beginning, I think you directly combine the bacteria and the polyphenol compound. I think it's very high risk, and it can inhibit the bacteria. I'm sorry because your presentation is very good, but tadi putus-putus, jadi so I cannot I cannot get it clearly. Okay, thank you, Saleha.
Okay, that's okay. Okay, no problem. So we use the skim milk as the vesicle to meet the bacteria and then the polyphenol extract. Okay, thank you, Irfan, for your interesting question. So I go back to the panelists.
Okay, okay, thank you, Kak. Okay, let's go to the second question to Kak Wildan.
Yeah, thank you, Rista, for giving me a chance to answer to responding the question from Mr. Irfan. As Mr. Irfan asked or get the questions about what is the most factor influence how the food will give the strongest umami taste. Is it correct, Mr. Irfan? That's a question, right?
Yeah.
Okay.
Actually, I'm sorry. Actually, what I want to ask is, you say that umami is actually not from glutamic acid, right? It's from 5 nucleotides and so on and so on.
It's not. No, I didn't mention like that. Umami is not merely. It's not merely. It means it's not always from the glutamic acid, but several compounds also can give the umami taste to the product, such as peptides, small peptides, and then nucleotides, any else.
Okay. I'm sorry. I got the wrong information. I think the umami is a combination of glutamic acid and another compound. So I want to maybe change a little about my question. I want to ask from that chemical compound, what is the chemical that have a biggest effect for us to test the umami, or there are out of their compound is have a same effect for us to test the umami or not?
Okay. So the questions is, sorry, would you like to repeat the questions for one more
Okay.
time?
Okay. So you say that we can test umami not from glutamic acid, but from another source such as 5 nucleotides and so on. And so from so many chemical compound that we can consume and we can feel umami, we can test umami from it. What is the strongest chemical that can make us feel umami?
Okay.
Do you understand my
Yeah,
question? Okay.
I get the question. I get the point of your questions. All right. Based on the previous research, state that generally or dominantly, food which contain umami taste is the food um, which contain uh, glutamic acids or several peptides. And generally, the peptides or the amino acid itself, such as glutamic acids, which is compound that has the molecular weight under 3 kilodaltons. That's why we use ultrafiltration process using the membrane 3 kilodaltons. So as higher compounds that has molecular weight less than 3 kilodaltons, as the strongest the umami test will be tested by the consumer. Do you get the point?
Yeah, but what is the chemical compound that have the strongest umami test?
Oh, we
Do
cannot.
you know? Do you know? Or from the previous research, do you know what is the chemical compound that have a strongest effect for us to test the umami? Not from your research, from the previous
Previous reason.
studies
Yeah,
or research. Yeah.
that's why we need the panelist selections, or we need a TDA or uh, test dilution factor to know which one the strongest umami of the food, which uh, I mean, not from the food, but the fractions, from the fractions of the identification using chromatography and using ultrafiltrations. So we need the panelist selections. We need the TDA test. It's not about how much. Uh, do you get the point of my response?
Yes. Just continue. Just continue. I know.
Yeah,
I get the point.
that's why uh, we need such a panelist selection. So combination between instrument analysis and panelist selections, or the analysis using the panelists will give the result whether the fractions contain the strongest umami or not.
Okay, I get the point of your answer, and I think it's enough from me. Thank you, Mr. Well done.
Yeah.
Okay. Thank you for Kak Wildan and Kak Saleha for the answer. Okay, let's go. Maybe anyone who wants to ask or give feedback, discussion, and many others, please raise your hand. And we can, yeah. Oh, Kak, yeah. There is one question, Kak Ayu Cahyani.
Okay,
Hello,
thank
Kak
you.
Ayu.
Yeah, I have two questions for Mrs. Saleha. The first one is, one of the goals of this research is Lactobacillus rhamnosus, to analyze the polyphenol antioxidant, but I didn't see on the slide the result of that. Could you please explain us? And the second one is, how is the process of rejuvenation of the Lactobacillus rhamnosus? Okay, that's for Mrs. Saleha. And the next one is for Mr. Wildan. One of the objectives of your research is uh, characterizing water-soluble extract, or WSE, of terasi that is potent in giving strongest umami taste. How to characterize these water soluble extracts? I mean the process. Thank you.
Okay, thank you Kak Ayu for the question. Maybe from Kak Saleha can start the answer.
Okay. Sorry, maybe Mas Wildan able to answer the question first.
Yeah. Okay. Thank you for Rista as the panelist, and thank you for Raden Ayu Cahyaning Alam for giving me questions regarding the objective of the research. That is water soluble extract to characterize water soluble extract of the sample. How to characterize this the water soluble extract that I mentioned before? There is analysis using LC-SEMS. The analysis using LC-SEMS is to know to characterize what kind of the peptide that give the strongest umami of the water soluble extract itself that has chance to be several fractions. After we know the kind of the peptides from the LC-SEMS instrument as the final identifications, we will have the data, and the data will be collected as one of the alternative sources beside the monosodium glutamate or glutamic acid, as generally it is used for the flavor, for the taste, for the umami enhancer or umami flavor into the food. So that that is the way how to characterize. So we use the instrument LC-SEMS to characterize peptide, because the result of the analysis is data in the instruments that is connected to the applications, that is connected to the computer or to the laptop using the application, specific applications. Do you get the point of my response?
Yeah, I guess. Thank you, Mr. Wildan. It's clear.
Thank you.
Okay, hello. Can you hear me? Hello.
Yeah.
Yeah.
Okay, I'm sorry, I lost my connection. Is it clear, Kak Ayu, for the answer from Kak
Yes,
Wildan?
I got it. Thank you.
Okay, thank you. Now, Kak Saleha ya.
Okay.
Yeah.
Okay, thanks for the interesting question, Ayu. I would like to answer it. And the first one is why I didn't give the result of the antioxidant, because of we now on the progress on published paper. So you able to see on my publication paper if is it done. So now is it in progress. Then I just give the result of the uric acid levels. So it's maybe it could make you too curious about my research, so you want to wait to my published paper. And the second question is what the method that I use for rejuvenation. I remember that I used the I don't know it is the method for rejuvenate or not, because when I accept the bacteria, the Lactobacillus rhamnosus bacteria, we did the we calculate the bacteria. So we use the aseptic technique method. So we use the total plate method. We calculate the amount of the bacteria. So we use the bacteria from the plate, from that plate. something like that. So I would say that from the bacteria, then I grew the bacteria first, and then I used the growth of bacteria. So I didn't use directly from the cellar. Something like that. Thank you. So I go back to the panelist. Is it clear?
Okay. Is it clear, Kak
Yeah.
Ayu?
It is clear enough for my second question, but for my first question, I'm still curious because you put the result of the uric acid, but you don't give us about the. Could you please give us a little bit description of your main result about the antioxidant? A little bit
No.
maybe.
I'm sorry, no, because you have to read my published paper, so it will make you curious. I'm sorry. So wait, our published paper ya.
Unfortunately ya.
Yes, buat sekarang ini aku ngasih yang uric acid levels aja. Jadi kalau misalkan if you curious on if you want to know more about my research information, you are able to access the published paper that now is going on. Hehehe. Makasih Mbak Ayu. But but I wanted to give you the sneak peek that the polyphenol extract, I mean like the concentration of the polyphenol extracts is support to lowering the uric acid. So the content of the extract of polyphenol is support the result. Jadi sinergis gitu. Cuman I can give the data karena ya you have to read our published paper. Terima kasih.
Okay, thank you, Kak Saleha. Yeah, maybe we can wait from the journal that will be published. Maybe you can notify Kak Ayu, Kak, if it had published. Okay, let's go. Maybe it will be the last question. Maybe the third question. Kak Pinondang ya. Kak Pinondang want to ask question, Kak?
Yeah, I want to ask a question to Saleha.
Okay. Okay, yes, please, Kak.
Okay. We know to make yogurt use Lactobacillus bulgarius, and your penelitian use Lactobacillus blah blah blah. I forget.
Rhamnosus.
Rhamnosus. Why you choose your bacteria Rhamnosus? What is the advantage your bacteria from the another? Thank you.
Okay, thanks for the question, Pinondang. Prior research said that the Lactobacillus rhamnosus is proof to lowering the uric acid. So the isolate of a Lactobacillus rhamnosus that give to the rats directly, it's able to lowering the uric acid. So that's why I use the Lactobacillus rhamnosus. In this research, I just want to combine the goodness of Lactobacillus rhamnosus to lowering uric acid, and also the polyphenol greens extract to lowering the uric acid. So I just want to combine those materials. Is it clear? Hello?
Punya perbedaan nggak ya Kak? Have difference hasil.
Yeah.
Maybe the characteristic yogurt.
Okay. In this research, I didn't compare the commercial yogurt with my own yogurt. I just look and get to know about my yogurt. But from the bacteria, it should be different because different bacteria, different content of yogurt, right? But in the sense, I didn't make the comparison about the commercial yogurt and then my own yogurt. But I guess it would be different. Of course, yes.
Thank you.
Gitu deh. Okay, thank you.
Okay, is it enough, Kak Pinondang? Is it clear ya?
Yes.
Okay, thank you so much, Kak. Maybe we can get one more question, maybe, for Kak Wildan. If anyone wants to ask Kak Wildan, is there anyone to ask? Please raise your hand. Oh, Kak Ayu. Kak Ayu come again. Okay, Kak Ayu. Please.
Okay, I just have simple question for Mr. Wildan. What is the most difficult process that you felt to conduct this research? Thank you.
Thank you. Thank you. I for really interesting questions. Actually, I haven't started yet the research until now because, as I mentioned before, the research will be conducted on March to is about June 2021. Perhaps we are facing the pandemic. The difficult one is will be in the panelist selections because we need is about 50 panelist selections, and generally the researchers about Umami they use they utilize students. Yeah, they utilize students for the panelists for the candidate of the panelists, which is 50 panelists. That will be a difficult or a trouble in conducting the research. And the solution is we will another alternative to conducting this research, which is specialization in panelist selections. We will use trained panelists, maybe in the laboratory or in the balai that they have the panelists. So we don't have to have 50 people as a candidate panelist. So we will use straight away the trained panelists. Do you get a point of my questions?
Yeah, I got it.
Yeah,
I pray that you will do your conduct good.
Yeah, because the panelist selection or the TDA, test dilution analysis, is one of the most important steps in conducting this research. It will be combined to another analysis to get the result of the Umami research itself. Without the TDA or without the test dilution analysis, we will have no result. Which one the peptides or which one the glutamic amino acid that contribute giving the Umami test to the product. Okay, thank you, Ayu.
Yeah, good luck, Mr. Wildan. Thank you.
Thank you so much.
Okay, thank you so much, Kak Ayu and Kak Wildan. I think it is last question because the time is up. Before we close the presentation, I would like to remind you that there is an absence and the evaluation form in the chat box. So you can fill. If you haven't filled, please feel to give to us some evaluation feedbacks maybe, and we will make another scientific presentation. Inshallah, scientific presentation fall 3 will be held in the 4th week in this month. So we will waiting for you to join as a presenter or as a participant. Please fill the presenter form that will regularly share to the in a group. Yeah. Okay. Maybe we can stop the presentation. We can end the presentation. Before we close, maybe you can open the camera. We make photo we make photo? Please, if you can open the camera and
Okay, please,
Yeah.
anyone?
Can capture memories. Yeah.
Barista, sorry, you able to turn on your camera
I think.
just for a second.
Okay. Hello.
Yeah. It's still good.
Okay, 1, 2, 3, smile. Okay, once again. 1, 2, 3, smile. Okay, thank you.
Okay. Thank you so much, Kak Pinondang, for the help. Okay. I think it's enough. I'm Rita as a panelist. I'm so sorry for more troubles today, and I hope you can still join SP in the next volume. Okay, thank you so much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Rista.